Today we will be focusing on transcription, part of the central dogma, DNA transcribed to RNA and translated to protein. We are finally done modifying the mRNA with capping and splicing, but we are still missing one more piece of the puzzle, which is tailing, before we can move on to translation. First off, in transcriptional termination, RNA polymerase is still chugging along and transcribing off of DNA to make mRNA. Encoded with the DNA sequence are these specific consensus sequences that act as little signals to the RNA polymerase that it is time to end the mRNA transcript. There are three important consensus sequences: the hexameric region, which is AU-rich, the middle CA sequence, and the U-rich sequence. Once these three consensus sequences have been transcribed, they will act as landing pads for tailing proteins. Tailing proteins are found riding on the C-terminal domain of RNA polymerase and are always on the lookout for these consensus sequences. The two tailing proteins that we will focus on today are named CPSF and CTSF. They have been on the C-terminal domain the whole time, but for the purpose of this video, we will introduce them now. As soon as each of their corresponding mRNA consensus sequences are transcribed, 5' to 3' by RNA polymerase, they will hop over to the mRNA transcript. CPSF will land and bind on the hexameric region, and CSTF will land and bind on the U-rich region. Then they will work together to call over termination factors and other proteins. One of these termination factors is an RNA endonuclease that will cut the mRNA at the middle CA sequence on the 3' end to free it from the RNA polymerase. But wait, we aren't done yet! At this stage, the mRNA is very unstable because it is missing its final modification step known as tailing. Poly-A polymerase, which is a very special enzyme that does not require a template, has also been called over to the scene to help. Poly-A polymerase will string together adenine ribonucleotides only on the 3' end of the mRNA. Poly-A polymerase is not alone. Poly-A binding proteins will come in and bind every 10 to 12 nucleotides. Poly-A polymerase will keep adding adenine nucleotides. Another, and another, and another, and another, and another. I think you get the picture. When there are around 200 adenine nucleotides, we have completed the poly-A tail. As poly-A binding proteins accumulate, they regulate the length of the poly-A tail by signaling poly-A polymerase to slow down until poly-A polymerase, along with the poly-A binding proteins, eventually leave the mRNA. The mRNA is no longer unstable and is officially mature and ready to move on to bigger and better things. Translation time! And Christmas time! Merry Christmas and a happy mRNA transcript!